these are the highlights of the conference on metabolomics, where we have presented some of the research we do in with several colleagues, and this is another poster I'm going to show as an example of metabolomic study and how it can be useful to understand different biological environments and clinical. Here will be more clinical, I think. So this is a study. It is led by Dr. Ramstedt, my boss, and several colleagues from Odessa University also participate in this because we cooperate around this study. One of the leading persons is Dmitry Sokol, and he's been doing big part of the experiments in this research. So the guy with the legs, I will tell you the story about this guy that has only legs. This is a person who potentially has some catheterization. So in different clinical conditions, people might have a urinary catheter installed. And the picture probably doesn't show all the details of the installation, but this is a less pleasant procedure both for men and women that includes planting the silicone tube up the urinary tract into the bladder. In case people cannot pee themselves, produce urine, then they have to have this catheter to be able to remove urine. Otherwise, the bad things happen, intoxication and so. But having catheter means having also risks for the urinary catheter-associated infections. And this is a leading cause of morbidity and also mortality, because if the infection is severe and people are with a compromised immunity, they might have sepsis based on the bacterial infection in the urinary tract. So this is actually a serious question. So people are trying to understand the infections in the urinary tracts associated with catheters. These are different types of bacteria causing it. The leading pathogen is Escherichia coli because, of course, it resides in the proximity. They reside in our guts, but when they end up in the urinary tract, they also might cause infection, not only colonization and infection. Pseudomonas aeruginosa is another pathogen that is often found in these kind of infections. So in this study, we look at at pairs of E. coli and Pseudomonas, two pathogens living together, because there is a number of situations when clinicians are trying to understand the infection, isolate bacteria, and then they see two types of bacteria. Who is causing infection? Why there are two there? Because you would expect urine to be sterile, sometimes colonized by Escherichia coli. But why two bacteria are there? How they're surviving there? Are they competing? Are they cooperating? That we don't know, and that might be important for treatment of such infection. And this project, Olena Rzhepishevska, is aiming to answer the question. So we designed some experiments to try to understand. First, we had pathogens isolated here on the plate. You see, this is UTI plates, and there are two types of bacteria growing. Red ones, these are Escherichia, E. coli. Blue ones, these are Pseudomonas aeruginosa. And this media is specifically designed so to be able to distinguish them for the colonization, for diagnostic of urinary tract infection. But we also use this media for viable counts to be able to quantify how many of these bacteria, how many of others. So these on plates, you can see them in the schematic picture of the urinary catheter, because they're building biofilms inside of the catheter and then blocking this catheter and spreading the infection. Okay, this is a scenario. The background. So Dmitry Sokol, he did these very valuable experiments, viable counts, on these UT plates, as I showed. And here, 1, 2, 3, 4 clinical pairs of E. coli and Pseudomonas were used. So if you look at this first panel, A, these are viable counts of Pseudomonas filled bars, and E. coli, these are dotted bars. And each of them were isolated together. So they're not just some kind of E. coli and Pseudomonas. They've been found in a single patient together. So we know that they can live together in a patient that has a catheter. And that's why we were interested in studying just specifically those isolates, because they are coming from the environment, which is interesting for us to study and to understand. And then the last one is PO1 and K12. PO1 is a Pseudomonas strain, and K12 and Escherichia coli lab strain. So these guys were used for the control. And so Dima calculated that in the media, which is called artificial urine media, the viable counts of what is it called, Pseudomonas, are higher in some cases, or equal to the Escherichia coli. If you look, the red bar is much higher, dramatically, several times the red couple than this blue one. The next one is 2. Oh, here. Do you see my arrow here? Arrow. No? Would you see that?
Yes, we can see it.
Okay. So you see this bar is higher than this one. And that means that it's more Pseudomonas in the biofilm, or when we analyze it with the viable counts. If we look at the next couple, again, more Pseudomonas. We look at this third one, again, more Pseudomonas. And we look at this, again, more Pseudomonas. And we look even at the control, more Pseudomonas. So basically, in all of them, in fact, Pseudomonas are taking over the consortium. They are more prevalent than Escherichia coli. This is in artificial urine media. When we're looking at the next panel, these are biofilms grown in isosensitive test. An isosensitive test is a media which is quite rich. It has a lot of components. If you look at the table 1 here, this is the summary of the AUM content, and then there is also modified AUM. Isosensitive test. Here, it has complex composition and also glucose. AUM media in its real recipe doesn't have any glucose. So if we look at the isosensitive test and biofilm grown in the isosensitive test, same strain all of a sudden behave differently, because all of a sudden Escherichia starts growing better. And if you see the first pair is the same, they're equal in their amount. In the second pair, Escherichia takes over, these green ones. Here is the same, and then in this purple pair, Escherichia takes over, but in the control, Pseudomonas grows better. But this is a lab control. So here we were comparing clinical strains with these lab controls, and well, it seems like lab control doesn't behave the same. But important conclusion from this experiment is that in different media with different composition, bacteria, Pseudomonas, and Escherichia grow to a different extent. Some of them perform better, and some of them worse. And Pseudomonas is clearly surviving better in artificial urine media, while Escherichia coli seems recovering when there is an isosensitive test. And looking at the composition of isosensitive test, we've been thinking that that could be due to the presence of glucose, because from the literature we know that preferred substrate for Escherichia coli, these are sugars. For Pseudomonas, the preferred substrate is a nutrient substrate, organic acids. amino acids too. But for Escherichia, these are glucose. So we were suspecting the glucose play a role here. That we prepared this other media, modified media where glucose was added. Also, we added some other components separately, but I'm not going to talk about them, only about glucose. So this media, aum, and aum with dextrose or glucose, was used in a very interesting experiment from my point of view that we found. It's called criss-cross experiment. Here's some typo, but the thing is that experiment goes that you start culture tube with E. coli, then filter bacteria away after a certain amount of time, in our case 3 hours. You filter bacteria away with a specific filter that removes bacteria, and then you add their Pseudomonas aeruginosa. So what we wanted to achieve with this, that Escherichia coli will start metabolizing something that exists in the media, and then you remove it, stop this metabolizing, add Pseudomonas, and see how Pseudomonas is using the products produced by E. coli. And then we do like this for 2 hours, and then we again filter away the bacteria, add the new bacteria, E. coli from the same, and these are done in couples, those clinical couples that I showed you, the pairs of Pseudomonas and E. coli. And finally, we culture till 20 hours and analyze the supernatant composition in all those tubes. So the hypothesis was that somehow they are using nutrients probably of each other or produce something that inhibit them. Could be. We didn't know what we were looking for, but the hypothesis was that they produce some metabolites that would somehow play a role in the interaction between these two species that are producing infection of the catheteres. Is it clear? Or should I clarify something on this slide? Because I know it's not so easy to probably get it. Do you have any questions?
Yes, I have a question. The final medium, which was incubated for 20 hours, it was without bacteria at all, right?
Yeah, in the end, we also we remove bacteria, of course, in the final stage, because we take only a sample without the bacteria. We just want to have supernatant. We remove bacteria by filter.
And what was the idea of incubation for 20 hours?
Yeah, E. coli is growing there for 20 hours till the end. So then we will have some final product.
And then it is removed, and then you analyze. Okay, thank you.
Yeah, we analyze all these tubes too. So we have like media without any bacteria, and it was aum, and then aum was dextrose, right? Then we add their E. coli, cultured for 3 hours, removed E. coli, added Pseudomonas aeruginosa, cultured for 2 hours, removed Pseudomonas aeruginosa, added again the same E. coli, and then cultured for 15 hours to a total time of culture, 20 hours, again filtered, and then analyzed with metabolomics. So basically, we let them, for example, I was eating a sandwich, biting it, and then I gave it to my cousin. And then my cousin started to eat it for 2 minutes. Then after 2 minutes, I said, give me back my sandwich, and I start eating again. And then this is what we want to see, how me and my cousin together eating this sandwich. But probably we're also breaking part of the sandwich and leaving it somewhere lying around. So we wanted to see how these two cousins are eating the sandwiches that we provide for them in two different medias. One aum, the other one aum supplemented with dextrose. This is like a messy picture, but I hope first you will focus on this a small one, because it's a analysis of the PCA. Actually, very strong separation for the PCA, principal component analysis of the samples of this experiment analyzed by metabolomics. So green one is the aum without any dextrose, and the blue one, aum with dextrose. Clearly, bacteria grew differently in that. And so then we plotted them separately. B, it is aum without dextrose, and C, aum with dextrose. And if you look at the colors mean the time. You remember we've been culturing first for 3 hours, then filtering, adding new bacteria, culturing another 2 hours, and then for 20. So yellow, blue, and red, these are the time points. The green one is the media at the starting point. And if we look at the graph on the panel B, then you can see the spread count towards to the left from media. Then the 3 hours blue samples come, then red, and the latest one, these are yellow ones. And PEP or EP means Pseudomonas, E. coli, Pseudomonas. This is a succession of the strains of the species in this sample. Anyway, we can see a trend in this graph going from media to the samples of 20 hours, and that's quite clear. But still, the separation here of Pseudomonas and E. coli goes a little bit up also, so we can go in two different directions. But it's different, both, despite that there is a certain separation here, it's still different from the graph C, where we can see a separation in the media with dextrose. And here clearly you see this group of yellow samples at the 20 point, Escherichia, Pseudomonas escherichia. So where the Escherichia coli was growing in the last moment, they're separating the most. There is something that separates them. And below here, D and E, these are respective loading plots to these score plots B and C up there. And if you look, how to read those plots, they're going together. The scores plots and the loadings plots should be read together if you want to understand what kind of metabolites are responsible for clustering. And if you look at the clustering of this Escherichia group, then it is sitting in the same place as the succinic acid, which is a metabolite probably responsible for separation of this group. And so we'll look more closely at the succinic acid in this experiment, plotted it. And so here, A, this is this crisscross experiment in the normal aum media without dextrose. And you see that succinic acid doesn't change so much. In some samples, probably it drops substantially, but still there is not so much change comparing to the start point of aum. However, in the graph B, here we see the growth of these bacteria in aum media with dextrose. And this plot has 3 parts. If you look down on the writing, this is PO1K12. This is strain 5 of Pseudomonas, and strain 13 of E. coli, and strain 6 of Pseudomonas, and strain 14 of E. coli. These are those pairs that I've been talking about. So here we have 3 pairs, and the columns are presenting the levels of succinic acid in the experiment crisscross, where we add, here we had first Pseudomonas, then added E. coli, then Pseudomonas again. This is the other way around, the pink ones. E. coli, then we add Pseudomonas, then again E. coli. And we see that in all those 3 pairs, in the last sample, where we add E. coli and wait up to 20 hours, the levels of succinic acid are rocket high. So his conclusion from this experiment is that E. coli is producing a lot of succinic acid. But how would we know if Pseudomonas in this situation making any use of succinic acid? So here down the next panel, it's a zoom picture of this picture Panel, which one? B, right. So why we need to zoom it? Because these succinic acids in the last point are so high, the levels are so high that you don't see the variation down there. But this variation exists. If we zoom it out, if we remove the last point, then we see probably not so much difference in this clinical strain, in the laboratory strain PO1 and CO12, but in two clinical strains, this one and this one, the clear increase of succinic acid uh, after cultivation for 5 hours. And when you add again Pseudomonas, it decreases. So what does it mean? Here was one level of succinic acid in the green one. After cultivation of E. coli, it increased. And after we removed E. coli and added Pseudomonas aeruginosa, it decreased. What would that mean? And the same situation in the next clinical strain. What do you think? Is anyone here who could tell me?
Please, dear participants, any ideas?
I would like to say again. So this is reflecting this experiment where we let one bacteria growing, removing it, then the other one, and then the first one again, like eating sandwich together. Anyway, my conclusion is, or our conclusion is like this, that when you add E. coli to this mixture with dextrose, they start producing succinic acid. And when you add Pseudomonas eats up the succinic acid, because I told you that organic acids are preferred substrate for Pseudomonas. This is like a general background that we know from before. But here we see it in the clinical situation that it happens. Pseudomonas, the E. coli produced succinic acid in the first step, and then when we added Pseudomonas, they added it up. And this is typical for the clinical strain. If we look at these lab strains, PO1 and K12, such a clear trend is not seen, even though we see a little bit of a decrease, a little bit increase, and a little bit of a decrease of succinic acid when you add Pseudomonas. But probably lab strains are not so good in this, but clinical strains are very good in this. So the conclusion that we made, at least at this stage, that in this crisscross cultivation, the succinic acid is produced from glucose in a womb. And Pseudomonas can consume this succinic acid later on. And a conclusion that we propose is that Pseudomonas and E. coli, they do not compete there. They cooperate. And they, since they have different nutrients of that they prefer, they cooperate around consumption of these nutrients. E. coli goes for glucose that it likes, but then it produces succinic acid from this, and Pseudomonas is consuming the succinic acid that's been produced by E. coli. So that's a kind of cooperation that we suspect here. They are using metabolites complementing each other instead of competing. And that the kind of strategy may provide them with survival benefits. And that's how this double culture or dual biofilms, as we call them, they can be more successful because they can utilize different types of substrate. And if you think that the glucose in the context of clinical settings, it could be people with diabetes, so it's not so strange that glucose will be found in the urine. It can be a real life situation, but we of course only proposing a hypothesis based on our laboratory experiments. So this is the experiment that I close with these conclusions, but then also I would like to thank our Swedish Metabolomics Center, which is at our university, where we do all our experiments. So all the experiments has been done there, and they're taking care of the instruments, and we just pay instrument time and use the instruments as a user club. But without this facility, of course, life would be quite complicated for us. So we're grateful to our colleagues. Another thing that I forgot to mention, I saw that some of you joined the Twitter already. Thank you. for this. One more thing that you can explore, and what I learned at the conference here, there is a picture from the conference. Jeff Sia from McGill University, he and his team are developers of this software, which is called Metaboanalyst. So if you go to their homepage, which is metaboanalyst.canada, you will find this very interesting website where there's a lot of tools. If you click on all these things, statistical analysis and pathway analysis, this is like if you're doing metabolomics, these are very interesting and useful tools, and the guys are developing them. I took a workshop on this, and I think it's very promising. Probably LCMS vector processing is a little bit too optimistic yet, but other tools are nice. And there's also tutorials, sorry, and this omics forum. So I recommend you to check this site too. That's it. Thank you for your attention and patience.
Thank you very much for such an interesting lecture. Please, dear participants, do you have any questions?
Yes, I have. Who first?
Hello.
Hello, Olena. Where's my question? Is it possible to use metabolomics in the same way as metagenomics for fingerprinting of some samples? I saw what it's possible on your presentation, but it's always necessary to cultivate microorganisms to increase the concentration and do high efficiency liquid chromatography, or like with sequencing, it's not necessary.
You know what, I'm sure it's possible and one can establish a pipeline, but I don't see the need for it, because you mean fingerprinting is like identification in the mixture, right? Metagenomics.
Yes, prediction of what type of metabolism of on this zone area. Maybe this is a water or soil, and you can predict what, oh, okay, in this area we have high level of plastic or something like this.
Yeah, I understand what you mean. Well, I think that if you talk about prediction, then of course it's genomics, because there you look at the genes and just predict what bacteria are capable with. Here, there would be a good validation, because metabolomics, as I showed in one of those previous slides before lunch, it shows you the snapshot, the moment where we are. Not like the capacity and potential of the bacteria consortia, but what are they doing right now? And this is valuable in itself. So you probably want to check your theory with the metagenomics. You say, okay, these guys, if we add them some plastic, they will produce this and this and that because they have this and these genes. But how to validate that, right? We're predicting it based on the genes, but we're not measuring the actual products of the gene activity. But if we do metabolomics, then we can validate the predictions. This would be a good way to use it, I would say.
Thank you.
Thank you. Any questions?
Thank you for the question. It's interesting.
Okay, I have a question. Do you have any statistical data which infections happen more often caused by a single species or by this dual infection in clinical?
You know, I can't like right now provide you with a statistic, but one thing with this urinary tract infection is that E. coli is found in urine very often, but it's not so that it's causing the infection. So E. coli is always the leading cause, as they say, of finding something in urine. This is always E. coli. But is it really causing the infection? Because from our experiments, we see that Pseudomonas is suited much better to survive in the urine. And it seems like one is like urine without any special additives, is Pseudomonas that is there. So I think it's a little bit in the shadow of prevalence of E. coli in this site of urinary tract. They can be there just colonizing without causing infection, but they're always majority. However, I will look up the literature, and till the next time we'll have a better answer.
Okay, thank you very much. So, dear participants, any other questions? Maybe you want to write something in the chat.
Yeah, I have a question. Do you hear me?
Yes.
Okay, super. Do you what do you think about to continue research quorum sensing behavior between Escherichia and Pseudomonas, or between others bacteria from urine? What do you think about that?
Yeah, quorum sensing is a very interesting topic, and absolutely, I'm sure that there should be some interaction on this level. There must be. But honestly speaking, I'm not the big expert of the quorum sensing. Of course, I see your point was using metabolomics because you can identify quorum sensing molecules. But this method that I'm using, I'm not sure it's showing the quorum sensing molecules so well. Then you need to have special targeted methods. And at this stage, I'm just trying together with Dmitro and Madeleine finalize the study. We added different substrates and see how bacteria behave there, only solely based on metabolism. Of course, I assume that general metabolism, central metabolism even interacts with the production of quorum sensing molecules. But that would be too far for us now to start working with that, because even these experiments are challenging. I mean, we will probably do it one time in life, but not in the near future. But I think that the point that you're raising here is very good.
Thank you a lot for answer.
Okay, more questions now? Olena, thank you very much for your will and strength to give us this wonderful lecture. We
Thank
hope
you
you
guys
get
for listening.
better. We hope
For
you
your
get
will and
better.
strength listening to me.
No, it was very interesting. So we hope you will get better as soon as possible. Okay. Dear participants, any other questions? We can let Olena go. I think we can let you go. Thank you one more time for joining us at our school. So thank you very much. And
see you. Bye-bye. Good luck.
See you. Bye-bye.